We discuss one of the very important topic of molecular biology, 5' capping and polyadenylation. Approximately one question every year asked in the CSIR-UGC NEET examination, particularly the June examination. They are asking, this is my observation. So if we have a good grip on this topic, we can certainly solve one question in the C section. So here we discuss the previous year's question. Approximately 10 questions are there, 7 to 10 questions are there. So we discuss one by one, and almost all of them are the C section questions. So the first question is 5', just a minute, yes. So the first question is the 5' cap of RNA is required for. See, a little bit introduction about the post-transcriptional modification. First of all, we discuss. So two different kind of modifications are there, right? Two modifications are there after transcription, right? After transcription, the first is 5' capping, okay, and second is 3' poly A tailing. Here we discuss two different kind of modifications, and another modification, third modification is also there, that is called splicing, right? We won't discuss splicing in this video, we'll discuss it separately. So here we focus on these two, 5' capping and 3' polyadenylation, right? So what happens actually when the RNA polymerase starts polymerizing, after initiation elongation comes, and in the elongation phase or during the elongation phase, once the mRNA has been synthesized, the processing also starts, right? The RNA processing, that means RNA modification, what we call. So RNA processing, RNA modification also starts, and first of all the 5' capping starts as soon as the transcript has been formed. The capping starts, and after the completion of synthesis of the mRNA, then 3' poly A tailing would be there, right? So here what happens, many other subsidiary enzymes are required, and the most important signal regarding the post-transcriptional modification is, like, this is the DNA, and here we are making RNA polymerase. Suppose this is the RNA polymerase, and this RNA polymerase is having a tail. See, this is the C-terminal tail, CTD, right? C-terminal domain, right? And once the phosphorylation takes place, the phosphorylation here, the phosphate groups added here, right? So after the phosphate group addition, they start recruiting other enzymes, and some of the important enzymes are here we write, and some of the important enzymes that we write here is the spt5, and another important enzyme which play an important role here is the TAT-SF1, right? TAT-SF1, right? And suppose this is the transcript, this is the transcript. Okay, this is the mRNA. Okay, this is mRNA. So here the function of the spt5 is to recruit the 5' capping enzymes, right? I mean enzymes which are responsible for the 5' capping, we'll discuss. So spt5, they help in the, or this protein will help in the capping, and TAT-SF1, this helps in the splicing, right? Just to keep in the mind, right? spt5 and TAT-SF1, right? And then we discuss 5' capping and 3' polyadenylation in the next slides. So here the question is, this is the June 2011 question. The 5' capping of RNA is required for, see, the capping mostly it occurs in the eukaryotes, of course, we know very well, right? Eukaryotes, prokaryotes, no such modification is there, right? So in eukaryotes, the thing is that entire transcription event occurs inside the nucleus, but we know the translation event that should occur in the cytoplasm. So the mRNA, if comes into the cytoplasm, then it gets many more challenges of different enzymes to degrade them. So to protect themselves, they modify themselves, right? So these modifications, they are useful to providing the stability of the RNA, right? Any RNA, mRNA, rRNA, tRNA, right? So they provide stability first, right? So the first is the stability, right? And the second important thing they help is the transport also, right? Right. So the second important thing which is important for the 5' capping is the transport also. So what happens actually in the 5' capping, they form a very unique kind of integration of guanosine residue, methylated guanosine residue, which is having 5' unusual phosphodiester bond. We'll discuss in the next slide. So here the answer would be the stability as well as the transport of the mRNA, right? So this would be the correct answer. All right. Next question is the eukaryotic mRNA are modified to possess a 5' cap, yes, 5' cap structure. Which one of the following is incorrect statement about the function of 5' cap structure, right? Incorrect. See, always be careful when they are asking incorrect statement, not correct statement, like that, right? I mean silly mistake, chances of silly mistakes are there. So which of the following is incorrect? See, 2019 में पूछा है, recently पूछा है, okay? तो incorrect कौन सा है? One by one we just go through. So it protects mRNA from 5' to 3' exoribonuclease attack. Yeah, of course. See, what happens? This is the mRNA. This is the mRNA, and as we discussed, this is the 5'. Suppose this is the 3'. So 5' capping here occurs, right? 5' capping. So this capping, it protects the mRNA from the different exonucleases, which keeps the RNA from 5' to 3' direction. Of course, this is correct. So this statement is correct, right? Second statement, it facilitates splicing of nascent transcript. See, many reports are there that 5' capping, it also helps in the another post-transcriptional event, that is the splicing, right? So this 5' capping, and 5' capping starts just after the formation of transcript. जैसे ही mRNA बना, कैपिंग तुरंत ही हो जाएगी यूकैरियोट्स में, right? So the 5' capping, apart from the protection from the exoribonucleases, it also facilitates the splicing of the nascent transcript. So this is also correct statement. Okay. Third is, it protects the transcript from the degradation of the RNase 3. See, this RNase 3 is the endonuclease, first thing. Endonuclease, right? And the capping, it won't protect the RNA from the endonucleases, right? From the exonucleases it protects the mRNA, but from the endonucleases it won't. And this RNase 3, this family of the enzyme, it cleaves the double-stranded RNA, right? ये double-stranded RNA को or endonuclease, right? It will make a cut in between the mRNA, any RNA, not RNA only, mRNA only, right? So this seems wrong, right? But we have to read all the choices. Fourth is, it facilitates the attachment of 14S subunit of the ribosome. Yes, of course. See, the transcription or post-transcriptional modification, that helps in the translation process also, right? So after the complete transcription, then translation occurs, and in the translation, as we know, the ribosomes, which will bind to the SD sequence of the mRNA, or the places which is meant for the binding of the ribosome. So this 5' capping, it also facilitates the binding of 40S ribosome. So here the correct answer is 3. Alright, because they are asking incorrect statement. Okay, they are asking incorrect statement, so our answer would be 3. Alright. Okay, if you have any question, you can write me in the comment box, chat box. Okay. See, this is very nice question. जून 2017 में ये पूछा हुआ क्वेश्चन है. इसके पहले थोड़ा हम वर्कआउट करेंगे. पहले जस्ट we go through the question first. So eukaryotic mRNA have an enzymatic appended cap structure consisting of 7-methyl guanosine residue, right? This is called 7-mg cap. Okay, we discuss now. Joined to the 5' initial 5' nucleotides to the transcript. Given below are few statements regarding the capping. And see, the strategy is quickly you read the last statement of the question, so that you can save your time in the CSIR-UGC NET or any kind of competitive exam. So ultimately they are asking which is incorrect. So you keep in the mind, right? And then you read the statements. Okay. They are asking incorrect statement. ये कौन सा सही नहीं है, है ना? Just a minute. Okay. So now the first statement is, the capping protects the mRNA from degradation by 5' exoribonuclease. Yeah, exoribonuclease they can protect, of course. Exoribonuclease se protect karega. Okay, so this seems correct. Okay, then second is, during capping, the alpha phosphate is released from the 5' end of the nascent mRNA. We discuss here. I mean, अभी हम थोड़ी देर बाद इसको pictorial representation एक करेंगे, then we discuss. So here they are saying alpha alpha phosphate is released, right, during the capping. Third statement is, phosphorylation mediated conformational change in the carboxyl terminus domain CTD of RNA polymerase 2 enables its binding to the capping enzyme. We again discuss here. Then capping, during the capping, 5' triphosphate bond is formed between the beta phosphate and the nascent mRNA and alpha phosphate of GTP. We discuss again. See, now we discuss the entire scenario here. What exactly the transcription, I mean post-transcriptional modification is. I mean how the post-transcriptional modification occurs. So here the 5' capping, 5' capping is of course the enzyme mediated process, right? This is enzyme mediated process, and this enzyme mediated process is completed in 3 different steps. 3 अलग अलग steps में होती है, right? And 5' capping. The first step is the removal of the phosphate group from the 5' end of the nascent transcript, right? So just we are writing here removal of 5' phosphate from the transcript we are writing, right? Transcript. Okay, first. The second event which occurs in the capping is the guanosine monophosphate moiety addition, GMP addition, है ना? GMP addition. And third is the addition of the methyl group, right? Addition of methyl group, right? CH3 group, right? 3 अलग अलग phases में है. हम next slide में देखते हैं and then we come back, right? So see, here I'm making a primary transcript first. Suppose this is the mRNA. एक just हम यहाँ पे rough diagram mRNA की बना रहे हैं. Right, this is the phosphate and this is the यहाँ पे जोड़ दो. Okay, and this is phosphate, right? ये चलो, this is the 3' end. Here the base is there, nitrogenous base. Here also the base is there, nitrogenous base, right? Here the phosphate, right? And you can make another nucleotide here, and this one like this. This is phosphate, and here you add adenine, because the first base which is there in the mRNA in most of the cases is the adenine, right? Okay, yeah, so you like that, right? So this is adenine, right? And here this is phosphate, 1 phosphate, 2 phosphate, and 3 phosphate, right? And we know very well this is 5' end. You can say 5' end. This is alpha, this is beta, and this is gamma phosphate. Okay, ये 3 phosphate इस तरह से. And now as we know, the addition of the GMP would be there. तो पहले क्या होगा? Removal of the phosphate, right? So we just write first the guanosine residue, right? पहले हम यहाँ पे GMP को ले लें. This is the phosphate, right? This is GMP, right? ये यहाँ पे आप दे दो OH, and this is base, nitrogenous base, और nitrogenous base में हम ले लेते हैं यहाँ पे guanosine, right? This is guanine. Okay, so again here this is alpha, this is beta, and this is gamma, right? So now what happens? This is the substrate actually. Now in the first reaction, the enzyme which is called RNA triphosphatase, right? RNA triphosphatase, phosphatase comes, and this RNA triphosphatase, what it do, it removes the gamma phosphate. किसको remove करता है? Gamma phosphate. It removes the gamma phosphate from here, right? We after discussion we then read the question again. तो यहाँ पे गैमा फॉस्फेट रिमूव होता है, ना कि अल्फा। वहाँ पे एक चॉइस में था अल्फा एक स्टेटमेंट में, राइट। सो दिस गैमा फॉस्फेट हैज बीन रिमूव्ड, and this after the removal of this gamma phosphate, this beta phosphate, right, ये हम लिख रहे हैं सेकेंड इवेंट की बात, इसके बाद जो हो रहा है। सो दिस gamma phosphate will attack onto the alpha phosphate of the GMP or guanosine triphosphate, GTP we can say, right। अल्फा पे करेगा, अल्फा पे करेगा तो क्या होगा, यहां से जो ये बीटा एंड गैमा है ना, this beta and gamma will remove it out, ये बाहर निकल जाएगा, this will go off, right, ये बाहर निकल जाएगा, ओके। तो यहाँ पे resulting bond, the resulting bond will form between the beta, this beta and this alpha, right, beta of the primary transcript है ना, ये mRNA है, ये primary transcript है, ओके। तो इन दोनों के बीच एक बॉन्ड बनेगा, वी आर, तो थर्ड स्टेप होगा, राइट, सेकंड स्टेप होगा। चलो सेकंड स्टेप में नाउ वी ट्राई टू मेक द दिस इज फॉस्फेट, देन अगेन, राइट, यहाँ पे जोड़ दो, हियर इट इज बेस, हियर इट इज एडेनिन, राइट, एडिनोसिन यहाँ पे डाल दो, एंड देन अगेन यहाँ से फॉस्फेट, राइट, एंड here this phosphate will join, here also the base is there, right, and this is, here this one is phosphate, this is alpha phosphate, this is phosphate, this is sorry, this is phosphate, so this is beta phosphate, ये beta phosphate हुआ, and then this will join, this is the alpha, this is alpha of the GTP, and now we can call GMP, right। so this, and यहाँ पे आप ये बना दो like this, and this is joined through here, and this is guanosine, right, okay। so and this is OH, so here the bond is formed between the alpha and beta, alpha of the GTP or GMP, whatever, right, and here the beta of primary transcript, right। so this bond is 5' 5' bond actually, right, this is very unusual bond, so most of the exonucleases, they confuse to cut this bond, they won't be able to recognize this kind of bond, so they are protected, I mean mRNA or primary transcript has been protected from the cleavage, right। so this is second step। so after that the third thing is there, that is carried out by another enzyme that is called guanyl transferase, right, the enzyme is guanyl transferase, right। so what the okay, so this guanyl transferase actually that makes the attack, I mean that helps in this attack, right, here we can write, not here actually, this is wrong, right, because bond has already been formed, so here the another enzyme is guanyl transferase, right, because first of all this alpha phosphate has been removed by the RNA triphosphatase, right, and after removal, this may be the first step, then this is second step, right, and then guanyl transferase will transfer the guanyl group at the 5' end of the primary transcript, okay, and then third step will come, this is the third step suppose, right, and third step is carried out by another enzyme that is called methyl transferase, right, methyl transferase, right. so what they do, methyl transferase just transfers the methyl group onto the guanine, third position of the, I mean third position of the guanine residue. See, if you sketch the guanine here, complete structure of the guanine, so like that it would be like that, ये हुआ, right? So this is the third position, right? So here one methyl group has been transferred, CH3, right? So this step is carried out by another enzyme that is called methyl transferase. So it will transfer a methyl group at the third position, right? And finally this would be, this is called capped mRNA then. So now we come back onto the question, right? So question is, the capping protects the mRNA from degradation by 5' exoribonuclease. This is correct. During the capping, alpha phosphate is released from the 5' end of the nascent mRNA. No, this is not correct, right? This is not correct. कौन सा यहाँ पे जा रहा है? Gamma will be released out, right? Gamma will be released out. So here alpha. So this is the wrong statement, right? ये गलत हुआ. Third is phosphorylation mediated conformational change in carboxyl terminus domain CTD of RNA polymerase 2 enables its binding with the capping enzyme. Yes, earlier in the first question we discussed the C terminal domain phosphorylation. You come back here. Here the phosphorylation. This phosphorylation invites many more enzyme, which may be the capping enzyme or which may be the polyadenylation enzyme. So here this statement is also correct. Phosphorylation mediated conformational change. After phosphorylation, conformational change is there in the mRNA, in the RNA polymerase 2, right? And this phosphorylation takes place at the C terminal domain, tail like structure is there, right? So that enables the binding of the capping enzyme, right? So this is also correct. Okay. Third is, during the capping, a 5' triphosphate bond is formed. This is correct. Between the beta phosphate of the nascent mRNA and alpha phosphate of the GTP, right? So this is also correct. See, this is the alpha phosphate of the GMP and the beta phosphate of the nascent mRNA. So this is also correct, right? So I'm sorry. So here the first is correct, second is wrong, B is wrong, third is correct, and fourth is also correct. So which of the following is incorrect? So only B is incorrect, right? This one. So B number choice, 2 number choice is the correct choice here. This is a good question actually. Many more questions can be solved by having this concept. Okay. So please note it down. Third question. This is also a very nice question, and in December they asked. HeLa cell extract was used to study the transcription of a X gene having 6 introns. Okay. See, long questions are actually simple question. So this is also a very lengthy question, long question, but this is simple question. RNA polymerase 2 complex containing all associated proteins was isolated from actively transcribing system and subjected to the proteome analysis. All right. Result showed the presence of both splicing and capping enzyme in the complex. Okay, both are there. Splicing and capping enzymes are there. When transcription elongation was inhibited by some antibiotic, suppose here the transcription elongation is inhibited by the flavopiridol, polymerase complex contained only capping enzymes, right? Inhibited by when the transcription elongation was inhibited by some chemical, the polymerase complex that contains only capping enzyme, right? This is one thing. When the phosphorylation of the CTD of polymerase 2 was inhibited by kinase inhibitor, the complex contained neither splicing nor capping enzyme. All right, neither splicing nor capping enzyme, right? First case में क्या था? When the transcription elongation was inhibited by flavopiridol, polymerase complex contains only capping enzyme, right? केवल capping enzyme. But यहाँ पे जब कर रहा है तो both capping enzyme and splicing enzymes are. Okay. So from these results, following conclusions were made. right? And again, first of all, we read the last part of the question. So identify the correct set of conclusion. Correct पूछ रहा है इसमें ना, which of the following is the correct set of conclusion? So here, transcription of gene X is coupled to mRNA capping. Transcription of X gene. So X gene they have taken and 6 introns are there, right? And when they are treated with the flavoperidol, the polymerase complex contained only capping enzymes, right? Only capping enzymes are there. So the transcription of the X gene coupled with the, of course, yes, right? Of course, yes, when the elongation was inhibited, when they inhibit the elongation process, right? Like here, inhibited by the kinase. So both capping as well as the splicing enzymes are not found. So this suggests the transcription of X gene is always coupled with the mRNA capping, right? Here also, if they are treated with the flavoperidol, so they still have the capping enzymes, right? So both are the simultaneous phenomenon. So this is correct. Transcription elongation is coupled with splicing. This is also correct because second inhibitor, this says that the splicing enzymes has also been disappeared, right? Neither splicing enzymes or capping enzymes. So this is also correct, this statement. Third statement, phosphorylation of CTD is required for recruitment of the capping and splicing. Yes, of course. Earlier also we have had discussion that phosphorylation of the CTD is the main root cause of the elongation as well as the post-transcriptional modification. So CTD is required for the recruitment of the capping as well as the splicing. So this is also correct. Both capping and splicing of mRNA occurs simultaneously. No, this is wrong. See, because here when this antibiotic or this chemical has been added, only the splicing enzymes has not been obtained, right? But here when the kinase, the thing is that the both phenomenon are happening not simultaneously, but when the kinase inhibitor has been added, so this is the, this blocks the phosphorylation of the CTD, right? This is the very beginning or the initiation of the elongation as well as the modification, post-transcriptional modification, right? So here certainly the both capping as well as the splicing of the mRNA occurs simultaneously. This is the wrong statement actually. This is not correct, right? Otherwise, by treatment of both the chemical, whether this is kinase inhibitor or the flavoperidol, so both splicing as well as the capping enzymes has not been obtained in the extract, right? Because they are coming after the treatment of the different chemicals. That means both processes are not simultaneous. And actually this happens, first of all the capping occurs, after that splicing, and then tailing, right? After capping the tailing is there, and finally the splicing will be there, right? So this is the wrong statement, and here the correct set of conclusion is A, B and C. So A, B and C are correct. Alright, this is also good question, simple question but good question. Next question is the eukaryotic cell undergoing mRNA synthesis and processing was incubated with P32, right, labeled ATP, right? mRNA synthesis and processing के समय पे क्या कर रहा है, P32 लेवल ATP डाल दे रहा है with the level at the beta position. Okay, now you can remember the beta position, right? Labeled ATP with the level at the beta position पे क्या किया, labeling हो गई, right? Where do you think the radioactive isotope will appear in the mature mRNA? कहाँ पे ये आएगा, mature mRNA में कहाँ पे आएगा, right? This is simple question actually, right? तो पहला हम पढ़े P32 will not appear in the mature mRNA under any circumstances because beta and gamma phosphate are released during the transcription. See, yeah, this is correct, but not for the 5' end. Right, we have seen that at 5' end, I'm coming to the figure again, right? At the 5' end, this is 5' end, so 5' end, beta is still there, right? Beta is still there, isn't it? So this is wrong. So this statement is wrong. Second statement, phosphate group of the phosphodiester backbone of the mRNA will be uniformly labeled as only gamma phosphate are released. No. See, in between both beta and gamma will be released and only alpha phosphate will be there, right? जैसे इसको आप देखो, so these are the alpha phosphate actually. जो बीच में है वो क्या है? Alpha phosphate है. So only the alpha phosphate will remain there, right? So this is also wrong. P32 will appear at the 5' end of the mRNA if only it has A at its first nucleotide. This is correct, right? That is how the 5' capping occurs, right? We have seen. Then last choice is no P32 will appear in the mature mRNA because 5' terminal phosphate of the A residue will further be removed. No, this is also not correct. Okay, so the correct answer would be third. The third is the correct answer. There is no pen. This one. So this is the third. Third number is the correct answer, right? Okay, so this question can be solved only when you have this concept. Okay, so you please you remember this concept. You understand this concept. Okay, now this question 2013. This is also a very nice question. You are working with an in vitro eukaryotic transcription system, right? Which produced both capped and uncapped mRNAs, right? Capped RNA and uncapped mRNA are there in that eukaryotic transcription system. Okay. You incubated these mRNA with mammalian cell nuclear extract and then quantified the different products as shown below, right? Which one of the following graph is correctly represents the expected result, right? Which one is the correctly represents the expected result? So in the eukaryotic transcription process, both capped and uncapped mRNAs are there. दोनों हैं. And nuclear extract में उसे रख दिया. So what happens then? See, the symbols are, this is the pre-mRNA, this is spliced mRNA, this blocked line, and this entire black or filled line is the polyadenylated mRNA. Okay. Alright. And here the capped and uncapped is there in all the 4 graphs. Okay. Okay, this is a very good question, really very good question. Okay. So what happens when you talk about the capped pool, right? जो capped mRNA isolate किया, यदि उसकी हम बात करें तो there the pre-mRNA will be very very less. जो ये वाला है ना, this would be very very less. Pre-mRNA would be very very less. ये capping हो रही है वहाँ पे, so there is no issue, right? And if capping is, if capping and polyadenylation that occurs very smoothly, then splicing also occurs, of course, right? So in the capped condition, so the splicing is also very well, very well noticed. One can notice very well. So this splicing or spliced mRNA will be, or concentration would be high. Pre-mRNA would be low because the capping is there, right? And consequently the polyadenylation would be, the polyadenylated mRNA will also be more, right? This will also be more. So this is more, this is less, and these two are more in the capped condition, right? But if we are talking about uncapped condition, uncapping के समय की हम बात करें तो uncapping के समय क्या होगा? यदि capping नहीं हो रही है, so pre-mRNA would be more, pre-mRNA बहुत ज़्यादा होगी. समझना चीज़ों को. Pre-mRNA would be very very high. कब? Uncapped condition में. If the uncapping, capping mechanism is not working. So pre-mRNA is more. Okay, if pre-mRNA is more and no capping is there, right, no polyadenylation is there, so no capping की हम बात करें, अनकैप्ड है तो स्प्लाइसिंग बहुत कम होगी, क्योंकि हमने पढ़ा पीछे कि कैपिंग दे ट्रिगर्स द स्प्लाइसिंग मैकेनिज्म, राइट, स्प्लाइसिंग मैकेनिज्म को प्रमोट करता है, एज वेल एज द कैपिंग दे ऑल्सो प्रमोट्स द polyadenylation mechanism. So this will also be less. See, ये ज्यादा होगा तो ये कम होगी, right? This will less and this will also less, right? The pattern would be that. Okay. In the capped condition, यहाँ पे हम लिख दें, यदि कैपिंग की बात करें और यहाँ पे अनकैपिंग की बात करें, राइट, अनकैपिंग की बात करें, यहाँ पे हम ये दिस इज द फर्स्ट SS we suppose and this is third, right? So this is first, this is second and this is third. When first is the pre-mRNA, second is the spliced mRNA and third is the polyadenylated mRNA, right? So capped mRNA में if this is low, right, of course if नहीं, ये होगा, ये low होगा, capping यदि हो रही है, so this will be high and this will be also high, right? And uncapping के समय this will be very high, right? pre-mRNA, uncapped pre-mRNA we can say. एंड यदि कैपिंग नहीं हुई है तो स्प्लाइसिंग नहीं होगी, पॉलीएडेनाइलेशन नहीं होगी, तो दिस वुड बी लो, ऑलराइट, लाइक दैट. अब हम ग्राफ में ढूंढते हैं. सो दिस इज लो, हाई, हाई, दिस वुड बी करेक्ट फॉर कैपिंग, कैप्ड कंडीशन. एंड अनकैप्ड कंडीशन की हम बात करें, सो दिस इज हाई, दिस इज लो, दिस इज लो, दिस सीम्स करेक्ट, बट वी हैव टू go through the other choices also. So here the capped condition is very high pre-mRNA. No, this is wrong, right? ये entire wrong हो गया फिर. Okay, यहाँ पे देखें, so okay, this is correct, but this is pretty low actually. बहुत ज़्यादा ये low दे रहा है. So this is not very true, we can say. Okay, as compared to A number choice. Okay, here this is true, but this is not true. I mean, this spliced mRNA is not at such a high level, right? So this is wrong. And here this is low. Yes, this is correct. And this spliced mRNA is very low. This is not correct actually. Okay, this is not correct ultimately. So the first number choice is the correct choice. This is a good question. I hope you understood. So if the capping occurs, then only splicing and polyadenylated mRNA we get in high quantity. Otherwise, the uncapped pre-mRNA will be high in amount or concentration, whereas the spliced mRNA and polyadenylated mRNA will be very less in amount. Okay, I hope. Okay, Mr. Subham, how are you? Good. Please write the question number also. Yeah, you are correct. Okay, next is next question. This is asked in the 2015. Okay, first of all we go through the question. 3' end of the most eukaryotic mRNA is defined by the addition of polyetyl, yeah, of course, processing reaction called the polyadenylation. Information: the addition of polyetyl is carried out by the enzyme polymerase. Yes, given below are few statements. See, before reading the statements, we first read the final line of the question. Which of the following combination is true? They are asking true, right? Okay, so the polymerase is template independent enzyme. Okay, this is template independent enzyme. This is correct actually. How this is correct? Okay, we have something to write here so that we can get the point how it is correct. Okay, first of all we just go through figure, then only we can understand how this is, how the choices are. So see, I'm talking about here polyadenylation. I'm taking a new slide actually, then we again come back onto the question. Suppose this is the DNA, right, and this is double stranded DNA, and this we are taking RNA polymerase, right? एंड आरएनए पॉलीमरेज दे आर हैविंग टेल, ये एक टेल ले लो आप। हमने देखा कि जो फर्स्ट क्वेश्चन के लिए हमने फिगर बनाया था सेम फिगर है, फॉस्फोराइलेशन यहाँ पे हो गई, राइट। एंड फॉस्फोराइलेशन मोस्टली टेक्स प्लेस एट द सेरीन फिफ्थ, राइट, सेरीन फिफ्थ सेरीन पे नॉर्मली होती है ये, फिफ्थ सेरीन के बाद सेकंड फिफ्थ लाइक दैट, ओके। तो फिफ्थ पे, और हियर द एमआरएनए में दूसरे कलर से बना रहा हूँ, दिस इज रेड कलर, राइट, एंड दिस इज द प्राइमरी ट्रांसक्रिप्ट, ये एमआरएनए बना रहा हूँ मैं, राइट, दिस इज द mRNA, right. So mRNA, we have had discussion about the 5' capping, so this is 5' end, right. Now we are discussing the 3' and what happens with the 3' end. Yes, Chirayu Trivedi, good evening. How are you, Mr. Chirayu? After a long time, nice to see you. Okay, so here what happens when the transcription is almost at the end, right, almost at the end. So there is a sequence in the primary transcript that is called poly A signal sequence, right. This signal sequence poly A signal sequence होती है इसमें, right, mRNA के ऊपर या RNA के ऊपर हम यूं बोले, poly A sequence on the poly A signal sequence on the RNA, right. And here two enzymes will bind, right, जब end होने वाला होता है ट्रांसक्रिप्शन का, so one enzyme which is bind here and another enzyme which is bind here, and I'm writing like this, right, ये ऐसा मैं कर देता हूँ। See this blank enzyme, ये जो enzyme है one, ये first नाम दे दो and this is second नाम दे दो। जो first enzyme है and the second enzyme है, नाम मैं लिख देता हूँ, this is called CSTF, and second protein which is called CPSF, right, CPSF, okay. So both will bind here, CPSF and CSTF, right. And this CPSF and CSTF carry out the process to the next level, and here the CSTF will release, कौन सा यहाँ पर रिलीज हो रहा है, फर्स्ट नंबर CSTF विल रिलीज, एंड CSTF जैसे रिलीज होती है, क्लीवेज ऑफ द mRNA वुड बी देयर, mRNA यहाँ से कट हो जाता है, राइट. सो हियर क्लीवेज ऑफ RNA, राइट. सो क्लीवेज ऑफ RNA विल बी देयर. सो RNA क्लीवेज यदि हो गई तो नाउ वी हैव, ये समझो रेड से है, दिस इज द signal sequence, and here the only protein which is bind is the CPSF now, right, CPSF. And now this CPSF will recruit another enzyme which is called poly A polymerase. मैं यहाँ पे कुछ ऐसा symbol दे रहा हूँ, right, this is PAP, simply हम PAP लिख रहे हैं, and PAP by the use of ATP, ध्यान रखना क्या है यहाँ पे, ATP, simply ATP ले रहा है, right, with the use of ATP PAP, यहाँ पे PAP will bind at the 3' end. This is the CPSF. ये हमारा क्या है, CPSF. This is CPSF. This is PAP, right? So PAP they join adenine one by one in the template independent manner, right? Template independent manner में ये join करते चला जाता है, okay? So here what happens? They join the adenine residue, ये adenine, right, ये join करते हैं लगभग 200, right, 200 adenine will be added here, right, ये PAP है, right, this is PAP, right. So this will add in the template independent manner, right, approximately 200 adenine will be added. और कैसे, तो ATP से ये एडेनिन को लेता है, ATP से, ATP को ADP में हाइड्रोलाइज कर देता है, राइट, एंड दे इनकॉर्पोरेट एडेनिन इन द सिमिलर मैकेनिज्म, और बाय द सिमिलर मैकेनिज्म एज द आरएनए सिंथेसिस अकर्स. जैसे आरएनए सिंथेसिस होती है ना, जैसे यहाँ पे टेम्पलेट इंडिपेंडेंट मैनर में होता है. कैसा होता है, टेम्पलेट इंडिपेंडेंट मैनर में होती है, राइट. Now you come back to the question. So question is, yeah, the statements are poly A polymerase is template independent enzyme. This is correct. Poly A polymerase catalyzes the addition of AMP. See, addition of AMP from DATP. No, ये DATP देके यहाँ पे गलत कर दिया है ना. ये DATP नहीं होगी, simply ATP. Because the RNA process, हमने क्या बात करेगी, RNA synthesis की जो mechanism है, by the same mechanism they add the adenine residues. So this is wrong. Everything is correct. Only the DATP देने की वजह से ये choice गलत हो गया. Okay. So poly A polymerase is the RNA template dependent enzyme. No, wrong. Poly A polymerase catalyzes the addition of ADP from ATP to the 3' end of the mRNA. Poly A polymerase catalyzes the addition of ADP बोल रहा है. No, right? AMP add करता है. This will add AMP instead of ATP, है ना? ये भी गलत हो गया. Then E number, poly A polymerase catalyzes the addition of AMP from the ATP at the 3' end. This is correct. Okay. Next is next statement, poly A polymerase catalyzes the addition of AMP from DADP. No, this is also wrong. Okay. So correct choice is E. E number सही हुआ और A number सही हुआ, right? A and E. So this third number is the correct choice. Okay. So this is the last question actually. So I hope you understood the concept, and I hope it will help you a lot. If you think this is useful for you, you can forward to your colleagues also. And again we meet next Sunday with the new topic. Most probably we discuss the translation, we start the translation or something which is related with the mRNA editing, and then we proceed to the translation process. So thank you very much for watching this session. Thanks a lot.